Everyone, thank you so much for coming. We're going to wait a little bit to see if more people trickle in. Okay. Hello, everyone. Thank you guys so much for coming to our Girls Neuroscience Night. We're going to get started now.
So let's learn about Helyx. Helyx is the world's largest youth research community with over 2,500 members. Members can participate in STEM leadership, research, volunteering, and competition opportunities for free. Volunteer hours are also available. So Helyx is hosting this event and many more. So if you want to get involved in Helyx, you should join our Discord at helyxscience.org. Also, to get involved, you can start your own chapter or join an existing one. We have lots of things, including tutoring, hackathons, research programs, outreach, social media writing, and seminars.
Yeah, and this will be our Neuroscience Night. We host many Girls Science Nights. So please sign up for the other Girl Science Nights if you enjoy this one and you really want to see female scientists get represented in our meetings. We hope that it can inspire a lot of young scientists and just the general community. So yeah, the link is in the chat right now. to sign up for the other Girl Science Nights that we have planned. Today, obviously, it's neuroscience themed. We have two amazing speakers. Miss Sumia, I'm really sorry I can't pronounce your last name, and it's Gemma Deegan, and they're going to be talking about their work. And hopefully, we're gonna give them time to present, and we're going to open the ground for questions. So, any questions that you have, I'm sure our speakers would love to answer. So. And again, thank you guys so much for agreeing to speak at this event.
So our first speaker tonight is Sumia. She's an incoming activator at the Knowledge Society. She's an R&D intern at Apollo Neuro, co-founder at Revive, brain-computer interface developer, neuroscience and mental health researcher, and a women's advocate.
Yes, and we'll be giving like the speakers their own time to introduce themselves, but these were just the features we had. Miss Deegan is, she works at Neurophotometrics currently, which is a company which she'll elaborate more on, but basically helping science researchers conduct studies in like brain activity and such. Yes, and she hopes to pursue a PhD in neuroscience and psychiatric research. So without further ado, we will begin with our first speaker. It's Sumia, and she can share her screen. She has a.
Yeah, let me just take a second to share my screen. Okay, can you guys see that?
Yes.
Yeah.
Perfect. So I'll get started. So with over 86 billion neurons and over 100 trillion connections, the human brain still remains one of the biggest mysteries to this day. So there are so many layers to how we think, how we feel, how we communicate, how we move. So if you're as intrigued as I am, neuroscience is definitely something worth looking into. So before we dive into my story in this space, first of all, I'd like to thank Sarah and the team for having me and all of you for showing up tonight. Hopefully I give you something to take home, contextualize for yourselves to help navigate where you stand in this space. So without further ado, I'm Soumya, a rising senior in high school and activator at an accelerator program called the Knowledge Society, which I'll talk more on in a few moments. I'm a brain-computer interface, or BCI developer, a neuroscience researcher, mainly focused in the mental health space, and of course, a women's advocate. So before we get into my story, I'd like to let you know that most of the rapid changes or the rapid growth that I've seen in myself is compact within the past year. So this all started the beginning of last September 2019, when I was nothing special. I loved science, I was curious, I was interested in the ins and outs of the human body, biological systems, like how plants worked and all that. I also recognized that the most pressing problems, as well as the most complex problems, existed within the healthcare space. But my notion of how I would be able to invent or innovate and disrupt the healthcare space was very conventional. I thought I'd have to put myself through years and years of school and plenty of research before even exploring the space. Boy, was I wrong. And this realization came to me as I was exposed to my mentors, my network, etc., in the Knowledge Society, or TKS. So TKS exposed me to what the world's biggest problems looked like and the tools that we could use to tackle them in the future. So this entailed emerging technologies from AI to IoT to gene editing. But I was fortunate enough to come across something I really loved, something that really caught my eye. And that was, as I said, the brain-computer interface. So I know what you're thinking. It seems like a very foreign idea, very sci-fi-esque, and that was definitely what I was thinking when I encountered it too. But simply put, the brain-computer interface essentially leverages the electrical brain activity to control programs and machines in your environment. So with this was so new to me. I was challenged to look into challenging research papers, connect with experts in the field. With that, I was so motivated and inspired to start building my own projects. So this started with my brainwave-controlled game of Pong, brainwave-controlled calculator. And here's a quick snippet of how that came to be. So essentially, I leveraged my brainwave activity using the Muse headband, which consisted of EEG sensors, or electroencephalography. And so these sensors have high temporal resolution and are able to record the electrical activity coming from my head. So this data was then streamed to the Python script that I wrote via Bluetooth to dictate the changes in the Pong game. So there are distinct brainwave patterns attributed. So this was then attributed to the upward movement of the Pong paddle. Then my blink was also affiliated with a distinct crest in the brainwave activity. So this was then able to move my Pong paddle downward. So with this, the movements of the paddle in Pong were to be controlled solely using the variations in my brainwave activity without my hand. So I was able to receive the ball being sent back by the computer program. And I know what you're thinking. This seems a little trivial, and you're right. This is definitely the tip of the iceberg, considering how much potential this technology has. So I started thinking, okay, how can I leverage this technology to solve an existing problem that doesn't have an effective solution yet? So this got me thinking, and I realized that this kind of autonomy would be really practical for those with severe or many motor disabilities, especially those with locked-in syndrome. So I identified the problem and realized that these kinds of technologies could be leveraged to create a solution. So those with severe motor disabilities can now use their blinks or twitches or whatever it is to control programs like keyboards or machines in their external environment and gain a much improved sense of autonomy. So as Bill Gates said, we're changing the world with technology. And I truly believe that without solving a practical problem, there's no real use for the technology that we're developing and working on today. So with that, as I said, I started dabbling in the idea of a brainwave-controlled calculator, very similar to the brainwave-controlled keyboards that locked-in patients use. So with the distinct crests in brainwave activity, you could move from key to key in the calculator. And should you want to punch a value into the calculator, the user would essentially match their baseline or stare without blinking at the screen for a distinct time frame. And this would allow for the punching of the value into the calculator. So with this, I was inspired to think bigger. Where in neuroscience were there big problems that didn't have personalized or effective solutions yet? And I came across the idea of so many mental health conditions that were receiving universalized solutions. So for example, there are over 264 million individuals suffering from depression as of this year, and this number is only projected to grow. But there are only roughly 4 types of antidepressants in widespread use to tend to the needs of 264 million unique brain chemistries. So this didn't make sense to me. So I guess you could say I was naive enough, but also passionate enough to decide that I was going to start developing a personalized solution for depression. So this is where our moonshot idea, Revive, came from. That's where the Moonshot company we developed stemmed. So this essentially starts with your neurotransmitter levels getting measured through the Revive headband, very slick headband similar to the Muse headset, consisting of neurotransmitter sensors. So these levels, your neurotransmitter levels, are then sent to the Revive app on your phone, which is paired with the headband using Bluetooth connection. So then the machine learning model uses this data as input to determine the appropriate magnitude and duration of TDCS, or transcranial direct current stimulation, to deliver through the stimulators in that same headset. So the output is then sent to the headband, and the stimulation is delivered for the predetermined duration and with the right magnitude. So with that, as I said, this is only the tip of the iceberg. This technology has incredible potential, and there are many more companies working on BCI technology. So just to give you a gist of what the future may look like, there are actually algorithms today being built to convert distinct brainwave patterns into English words, spoken words. Along with EEG technology, there are a bunch of other neuroimaging techniques. So this neuroimaging and neurotech. So this includes the ECOG, or electrocorticography systems. And so these ECOG systems are now being leveraged to treat conditions like paralysis. So essentially, the difference between ECOG and EEG is the fact that ECOG is invasive, so a brain chip is inserted onto the cerebral cortex. And there is a link between this brain chip and an electrode at the end of your electrodes at the end of your spine, for instance. So someone who's paralyzed can think, okay, I'm going to move my leg, and this information is sent from the brain chip to that electrode to stimulate the muscle fibers in that inactive leg, and now they're able to employ that inactive leg. So this was actually a study that originally started with a research team in China who essentially did this with a paralyzed monkey, sending the information from the brain chip to the electrodes at the end of the spine to stimulate the activity in the legs. So this is incredible, but there's also opportunity for brain-to-brain communication over long distances and even downloading thoughts. So these are very foreign sci-fi-esque ideas, but this is what Neuralink, Elon Musk's company, is working on. Incredible. And there's also another common goal of achieving an everyday BCI to attend to tasks like as simple as activating your phone. And so companies like Neurable are working on this everyday commercialized BCI. So this is super interesting stuff, definitely something worth looking into. And with that, that essentially wraps up my technical journey in neuroscience, and I'm continuing to explore the potential intersections between neuroscience and other areas in healthcare. But a huge part of who I am today is also attributed to the 180 that my mindset did. And a lot of the guidance that I gained, definitely a lot of the credit goes to my mentors at TKS, my network, the support system I got. And so I thought I would share a lot of the valuable takeaways that I got, some things that you could contextualize for yourselves. So as we all know, it's common to see that girls are definitely underrepresented in STEM. And of course, this is changing, like initiatives like this. I think it's awesome. I'm so glad to see it. But when I started off, when I was encountered with these technologies, sciences, the research involved, I definitely felt intimidated. It could have been the imposter syndrome. It could have been that I was still yet to teach myself to code in new languages like Python, whatever it be. So I started off with a little bit of exposure to HTML, CSS, JavaScript, and that was about it. Then it was up to me to teach myself to code in Python, for example, to really dive into BCI development. So with that, I started realizing that most things can be taught. So then with the right intentions, frameworks, network, and of course, work ethic, I was able to really explore and find my niche in neuroscience and recognize which problems not enough people were working on and realize where I stood in the space. So with that, my idea of success started to shift too. Originally, as I said, I was very dependent on the conventional path to success. So then after I gained these skill sets and mindsets that top executives are employing today, I was empowered to start making an impact now. My dream of solving some of these extensive, complex problems in healthcare, that process was accelerated because I was capable of starting now. So I started to understand unconventional success. And I think that's so important that youth our age start to wrap our heads around. And another thing, as I said, with success, my outlook on failure also started to shift, considering this new personal mantra I adopted, and I'm so excited to share with you guys. It's the idea that I asked myself, do you feel the fear? Well, you're going to do it anyway. Feel the fear? Do it anyway. So by objectivizing the situation and really identifying what the worst case scenario was, I was able to gain a much more balanced risk assessment and realize that the worst case scenario really wasn't that bad, especially next to the potential reward, which included the lessons that come from making mistakes. So potential embarrassment, etc., associated with that worst case scenario be far outweighed by the potential reward. So finally, a key lesson was that there's something you can learn from everyone. So disregarding any biased first impressions you have of people, I think there's something so valuable that you don't have that you could potentially learn from every single person you meet. So it's about your perspective, what you focus on, whether you're focusing on the positives or those biased impressions. And this will allow you to really navigate which values you preach, what kinds of ambitious people you want to be affiliated with, and this will help you find the best mentors and support system for you. Trust me, this was life-changing. And this is something I continue to work on. So before we wrap up my story and my spiel of how I did in neuroscience and what I continue to explore, I want to leave something actionable for you guys to take home. So firstly, as I said, go out on a whim, start exploring. This is definitely something I take pride in, considering if I want something, I won't think twice. I'm quite impulsive. I'll jump right in to see how I feel about it. So if you like it, great. Start diving deeper and figuring out where you stand. Start reaching out to experts in the field. Start reaching out to people who have gone through similar processes to ask your questions and get those questions answered. If you don't, so what? Don't fret. Move on. I think it's so important as we do. This is an idea that we preach at TKS too. It's the idea that it's important to develop a breadth of knowledge and then find the areas in which you can dive deep, a couple areas. So I think that's so important for you to start exploring so that you can start navigating your niche and start realizing what your passions are, what you care about, what problems you want to solve, and really finding your network and your support system. So another thing is to put yourself on the line to find those people. You have to get comfortable with the discomfort of just reaching out and meeting new people, meeting strangers, cold emails, cold LinkedIn messages. But with that, you'll start realizing who those people with similar values are, similar ambitions. And it's so healthy, and I think it's a very sustainable way to develop relationships, considering you'll be able to help each other grow. It's life-changing. So with that, I'd also encourage you all to feel free to reach out to me. I'm happy to mentor people. I love meeting new people, gaining new perspectives. So don't hesitate to reach out if you want to discuss the technical sides of my work in the mental health space, the BCI tech I've been focusing on, or even just personal growth. Like I've experienced incredible changes over the past year, and I'd love to share. So here are my socials. Feel free to connect. With that, I'd like to give you guys 10 or 15 minutes to pick my brain. Shoot away. Ask any questions you have for me. Thank you.
Yeah, so any questions you have can be typed in the chat and we can read them out. One question I had was like just it's a bit more on the technical side, but what you said about the headband sensing the neurotransmitters, I was like really like, whoa, how is that even possible? Could you explain a bit more on how you can like sense neurotransmitters like just using a headband?
Yeah, absolutely. So, as I said, this is a moonshot idea, and so we're still working through some of the loopholes. And I definitely agree with you. It's definitely difficult because even things like portable MRIs are very difficult to scan through, like the layers in your head, right? The bones, the tissue. And I definitely agree with you. And so we're starting to think more about, okay, so the TDCS treatment is a practical thing, but it's the sensors that may be difficult, especially in a slick headset. So this may be something more clinical. So we just have to figure out where to pivot. So this is, yeah, you're 100% right. This is something we're working on in terms of, fleshing out the details. But yeah, it's definitely a challenge, especially to get through the layers and layers in your head to determine, like whether you have an influx or deficit of certain neurotransmitters affiliated with conditions like depression.
Well, sorry, another question that was in the chat was what women's advocating work do you do? Yeah.
Yeah, so I think my biggest role is encouraging, like what you guys are doing, encouraging girls' participation in STEM. So an initiative at TKS is called Boss Ladies to really push to create a tight-knit community for the girls working in STEM, working on big projects, working alongside men and whatnot. And so I think it's so important to create, I guess, creating the right network, very trustworthy network with other women in STEM, and also sharing opportunities to empower voices that usually don't get the light of day. So I've also recently been focusing on the intersections between neuroscience and things in women's health, for example. So things like postpartum depression. So if you guys have any questions on that, happy to answer. And another thing is things like leveraging these technologies to solve the root causes of things like sexual violence, rape culture. So that's another aspect of what I've started to dive into. So yeah.
I can read the next question. So the next question is, how do you advise we should approach cold emails? I want to go for it, but I want to make a great impression through the structure of my email/reaching out.
Sure. So this was definitely new to me too. When I started doing this, it was very experimental. But I think first of all, I'm sure you guys all hear this, but it's so important to really convey that you're passionate and show what you care about. Make sure you've done your research. Show that you know what you're talking about. And depending on who you're reaching out to, so if they're professors and you're looking for more, I don't know, research opportunities, start looking into their papers, start exploring what kind of research they've done, and explain why that's important to you, why that serves your goal. And another key thing is that you can't always say, I want this, this, and this from you. I think it's so important to share how you could add value to them. And so I think it's important to share your portfolio, things you've written, things you've read. And so, yeah, I think it's very important to find things you care about and show that you've done the work, even if you lack experience, to show that you've put in the work, preliminary steps to add some value to them and show that they have something to offer you. You have something to learn from them. So yeah, for me personally, I think LinkedIn has worked really well, but cold emailing is definitely a good option. Yeah, so if you want your like cold email template proofread, I'm happy to do so. I can share mine with you too. So yeah, don't hesitate. Like, I think that's another thing. Like, as I said, like if you feel that there might be there's a possibility that something might go wrong, that's even more the reason to do it, right? So I think it's so important to get used to failure too, and there's so many lessons that come with it.
Yeah. Someone else asked a question, but I'm gonna ask the next one because it's a bit more relevant. Like, how do you decide who to cold email so you don't waste your time writing so many emails to different people?
Sorry, is the question, how do you decide who to email to?
Yeah.
Okay. First of all, I definitely think you need to define what your goals are. Like, what do you want to get out of this? Is it a research opportunity? Is it just a connection? Do you need some technical questions answered? Whatever it is. And I think, as I said, it's just about who you think would be you could gain value from. So if you're interested in someone's work, if you have some questions to ask them, by all means, like shoot them a cold email and kind of have a couple questions that you would like answered. Like it definitely helps them to scope out that you care and also what kinds of things you're interested in so that they're well prepared to respond.
Cool. I can read the question that was before. Why did you choose neuroscience? Is it more interesting than other areas?
Yeah, that's an interesting question, actually. So I don't know. I guess I started thinking about I like how consciousness worked and how mental health conditions worked, like how anxiety and things like that. Things like um, how. So actually, originally I started looking into HIV/AIDS and how stimulation in the brain can actually help solve that. Obviously, that's another moonshot idea, but that's when I started realizing that there's so many layers to the brain and that there's so many questions that go unanswered. So if you guys start looking into connectomics, you'll understand that there's so many connections in the brain that are so difficult to map out, and people are still working on this. And so I guess it was just sheer curiosity and the idea that this 1.5 kilogram mass in your head is basically controlling everything, your thoughts, your feelings. And so I guess it was just sheer curiosity, and I realized that there were so many intersections between neuroscience and these other fields. So that's what fascinated me. And I would also say that that was a personal preference and just kind of how I ventured into the space. So I wouldn't um, disregard any other specialties or whatever it is, because I definitely continue to expand my breadth of knowledge. But yeah, I definitely think it's my niche, and I definitely care a lot about mental health conditions and figuring out how I can tackle like complex problems within the space.
Another question is, can you explain more what TKS, I think the Knowledge Society, is? I think the person may have not caught it in the beginning.
Yeah, absolutely. So TKS is essentially a accelerator program for youth, 13 to 17-year-olds, and exposes us to emerging technologies, AI, IoT, gene editing. I could go on, because these are critical tools to solve some of the biggest problems in the world, especially in with the future that we may see down the line. And another thing is that we're equipped with the mindsets of like the top executives that we see today. So for example, like the idea that most things can be taught was something I learned from Tom Bilyeu. Brilliant guy. Check out his podcast. I think that's another way to really get exposed to these ideas. But those are the two key things that set TKS apart from other, I guess, STEM programs. And it's so important to realize that the community in TKS is also very critical, and it's refreshing to have like-minded people and very ambitious people who understand what unconventional success looks like, and the opportunities and projects we work on are mind-blowing. Like over the past year, I've worked on a couple consulting projects with companies like Raven Telemetry. It's an AI company based in Ottawa, and Kidogo, a childcare company based in Kenya. So yeah, that's basically the gist of TKS. Incredible program. I definitely think it was life-changing for me. So
Cool. So our next question is, do you have any tips on how to be updated on latest technologies and science innovations?
yeah, I love this. Okay, so my first tip would be to join Slack channels or Discord channels or whatever it is that are interesting to you, because people share a lot of very cool news and updates and TED Talks or whatever it is. So I think that's a great way to learn. Another thing is set up your Google alerts and indicate like which words you want, like keywords of the topics you want to explore. And I think that has done wonders for me. Another thing is just, yeah, keeping up with science news, like reading up, connecting with people, staying up to date on, I guess, like big problems, big solutions that people are working on, and seeing, like where not enough people are working and figuring out how you could navigate your way into that space.
Well, thank you. Next is a pretty lengthy question, which I love, from Ms. Deegan, and she asks, to play devil's advocate and entertain the ideas of conspiracy theorists of the world, with the sophisticated cyber attacks that can hack some of the most secure internet networks in the world, what do you think is potential for the cortical chip you spoke about to be hacked with people with bad intentions? In other words, what do you think of the potential for mind control?
Yeah, absolutely. So this is something I get asked a lot, and this is actually something I was curious about when I got into BCI technology too, especially considering what Elon Musk is working on, like brain-to-brain technology. Like, think about it. If we're downloading thoughts, like it may seem very practical. I mean, you're saving a lot of time. It's very effective, but as you said, there are a lot of downsides to it. But to be totally honest, I don't know how this consequence would be dealt with. But as of now, I could definitely tell you that this is not something that we have to worry about within the next 5 or 10 years, because BCI technology is still in its preliminary stages, especially with invasive things like ECOG, like these brain chips. But yes, you're 100% right. And I think that's another big reason that would prevent the democratization of these kinds of brain chips. So yeah, it's definitely a very interesting topic, and a lot of the ethics of AI algorithms, BCI algorithms, are still a topic of discussion today. So
That's really cool. So our next question is, when did you start learning about computer science in general, and how did you find that neuroscience was the right area for you? Thank you for a great presentation.
yeah, thank you. Thank you for coming. So, okay, I'll be totally honest. Computer science is definitely something I'm not too passionate about. I'm definitely a science girl. Like, I'm very curious. I like the research side of things. But I think it's so important to realize that AI is so pervasive and so powerful. And so I realized that this technology, these kinds of emerging technologies, have so much potential to solve like prevalent problems in neurodegenerative diseases or the healthcare space. And so I thought, okay, I have to sit down, grit my teeth, and teach myself to code, like for example, like Python, to really get into BCI development. And so that's essentially my experience in computer science. It was through self-agency, and I was self-taught. And I'm still continuing to work on things. I'm no pro. But how I decided that neuroscience was for me, I guess it was more of a feeling thing, in the sense that I was very excited by it. I realized that there were so many complex problems that not enough people were working on. Things like I don't know, like solutions for the paralyzed, mental health conditions, depression, anxiety. Those are very sensitive and complex problems. So that was very interesting to me. But also the fact that it was almost like an outlet, in the sense that I would be doing like tedious work, like from school, whatever it is, and then when I come down and sit down just to read about like innovation and whatever's happening in neuroscience research, I was excited, and I could sit there for hours on end. So I thought, okay, this is definitely something I'm passionate about, and something that I could take further into my future. So I guess finding my purpose and passion in neuroscience kind of came with how I was feeling. If I didn't care for it, I kind of just moved on. So computer science was kind of like a foundational thing for me, to really get into, okay, like how can I solve these problems in neuroscience?
Yeah, and building off that question, the next question was, did you self-learn Python, and can you expand on how you learned the language?
Yeah, so I think I was super lucky in the sense that I had a bunch of friends who, these guys are super proficient in computer science. That's all they care about. They're working on AI algorithms, blah, blah, blah. And I'm like, okay, this is great. But they definitely had great recommendations for like crash courses. And so one thing I would recommend is the MOONSHOT course for Python. That's essentially what I use to learn the basics of Python. And then if I had any questions as I was writing the scripts for my BCI systems, for example, I would just look them up, or reach out to people in those Slack networks, on LinkedIn, whatever it is. But yeah, a lot of it came with self-agency, and I was kind of, I think another big mindset was being able to figure it out and kind of being more self-reliant. So whatever it is, without depending on other people. You'd have to ask the right questions, look in the right places to get your questions answered. So yeah, I am self-taught, and I'm still working on things. So I'm no pro when it comes to programming, but I think it was definitely foundational and very critical for me to dive into BCIs and the technology that comes with neuroscience.
Okay, so our next question is, which companies did you like working with best, and what projects did you do with them?
Yeah, so I've done a couple of consulting projects, as I said, but this summer I was able to intern with Apollo Neuro. And so this was super fascinating for me, as I said, first of all, because I would recommend looking into the work Dr. Rabin has done. Awesome guy, super cool. He's done a lot of stuff with psychedelics and like sound wave therapy with stress, and that's essentially what Apollo was built off of. So they're using wearable technology to mitigate the symptoms of anxiety and stress. So this kind of inaudible sound wave therapy suppresses the sympathetic nervous system, so that's your fight or flight response, and engages the parasympathetic nervous system, so that's your rest and digest. And so this is incredible. Like, it's a wearable, it almost looks like a watch. And another interesting thing is that I am a research and development intern with a couple other people, and we're being mentored by Dr. Rabin. And so we're essentially conducting our own research study in hopes of seeing any patterns in data between the use of the Muse headband and the Apollo Neuro at the same time. So this is new for me. We're still working on this. I'm super excited about it, considering it's very focused on biohacking, which is very interesting, something I've started looking into. So yeah, I would probably say that that's my favorite thing, considering it's kind of right up my alley, and it's more of like an independent research study. So we're building the platforms, we're building the like data collection systems, and we're asking the questions. And so I think that kind of self-agency, independence was really exciting for me, and obviously it's within neuroscience, so that was very exciting too.
Thank you so much. So Mia, hopefully I'm pronouncing that right.
Yeah.
I don't think we have any more questions. Oh, we have one more question. It asks, are there a lot of women in neuroscience at your workplace?
Like where I'm interning now, or?
I guess, yeah, however you want to interpret it.
Sure. So actually on my team, I'm the only girl, but there's only 4 of us, so including my mentor. So there's mentor, 2 other guys, and me. But that in itself is obviously not a good sample. Overall, a lot of the interns that came into Apollo Neuro, there's actually quite a good balance of girls working in marketing, working in research and development, whatever it is. So yeah, I would say generally people my age, people within the in interns, there's a good balance of girls. But yeah. Yeah, I can take that question too. So yeah, I'll tell you what I'm taking. So I'm in the IB program. I took bio and chem, and then I took physics in the summer because I was just curious. I definitely love the sciences, math, and I also wanted foundational knowledge. But yeah, I would recommend all the sciences. Take as many maths as you can. I definitely think statistics is very important, data analysis. And what else? Take other courses that you're passionate about. I think business is also a very useful thing to consider, especially if you're thinking about entrepreneurship. But yeah, also it gives you a good understanding of what the market looks like. So, sorry, I missed that last question.
Yeah, and just one last question. I think someone asked, what grades did you take bio and chem?
Yeah, okay. Hopefully I'm understanding your question correctly. But the IB program is 2 years, so grade 11 and 12, and I'm a rising senior, so I'm going into my second year of IB. But I did pre-IB for grade 9 and 10. Before then, I was just at any other middle school. So I guess you could say 2 years, because I started taking bio in grade 11, because that's when the separate science courses were offered to us. So yeah, hopefully that answers your question.
Yeah, thank you so much. I think that will be it before we move on to our next speaker, but thank you so much for your presentation. I definitely learned a lot and it was really motivating, and I'm sure it was the same for a lot of people in this call.
Yep, I'm in high school. Yeah, thank you so much. Thank you for having me. This is exciting. I'm glad you guys are doing this.
Thank you. Yes, and now we'll be moving on to our next speaker. It's Deegan, and she'll get to introduce herself, I'm sure, a lot better than I can. And she also has a presentation prepared for us.
Yeah, hello, good evening. As Chris said, my name is Gemma Deegan, and I'm really honored to get to speak to you this evening, and hopefully you'll get some value from my talk. So I'm going to try and figure out how to do this. Okay. Okay, here we go. So let's get started. Okay. I can't see myself, but I can see what I'm displaying at least. So hopefully that'll work for everybody else too. Yeah, so Chris very kindly asked me to speak about my neuroscience journey up to this point and where I currently work and what I currently do, and gave me pretty much free rein on what I wanted to base my talk on. So I'm going to do my best to convince you of why neuroscience is a really great area to get into in general, career-wise and research-wise. And yeah, hopefully it'll promote neuroscience. And I think what everybody's doing here is really important, especially for women in neuroscience and women in STEM. And any kind of promotion of the STEM subjects is always very, very welcomed. So great idea, and keep up the work. So as I'm sure my accent has so far indicated, I'm from Ireland. And my colleagues quite often tell me that I speak so fast they can't understand what I'm saying. So I'll do my best to try and speak nice and slowly so everyone can understand and keep up. And of course, we'll have the same question series at the end if it's a thing that I say anything and you miss it or you can't keep up. Great. So again, just to give a brief recap, just to bring everybody up to speed, I thought I'd give you a little introduction into what neuroscience is, since I'm sure there's a different kind of a varying range of understanding of neuroscience and the kind of areas that are available in that sense. So neuroscience is obviously the study of the nervous system, composed of the brain and the spinal cord, along with a network of peripheral nerves which run throughout your body. And these extend into your limbs and your organs. The brain acts as the control center and the relay center for all of these inputs and outputs from these nerves. And our nervous system allows us to move, to talk, to think, to breathe, hear, and see, as well as carrying out many other functions. So even when we aren't actively conscious, our brain is very, very busy. For example, when we're asleep, our brain is still quite active. So a neuron, which is a nerve cell, is the singular functional unit of the nervous system. And as was alluded to earlier on the previous talk, it is estimated that we have roughly around 86 billion neurons in our brain alone. So that gives you just a very rough indication of the amount of neural networks that overlap and that function together to ensure the correct functioning of our nervous system, both in our brain and throughout our body. Let's get these two bits. So neuroscientists study both how the nervous system functions optimally, so when it's behaving as it should be, as well as trying to determine how and why things don't work as planned during disease states. Some diseases that are widely studied in neuroscience include neurodegenerative diseases, such as depression or I'm skipping the line, such as Parkinson's or Alzheimer's, neuropsychiatric diseases such as depression or schizophrenia, and neurodevelopmental disorders such as autism or ADD, attention deficit disorder. And neuroscientists employ a wide range of approaches experimentally to their studies, with test subjects varying from humans to fruit flies to cell culture and everything in between. And finally, then there are many branches or subcategories within the field of neuroscience, which makes it very attractive if it's a thing that you're not solely interested on the cell aspect or the human aspect, or all those things. And these subcategories range from computational neuroscience, which uses mathematical and physical applications to model brain function or neuronal networks, to cognitive neuroscience, which essentially integrates the fields of psychology and psychiatry with neuroscience. So just a little bit, God, I realized the title for this is wrong, but just an introduction to me and my beginnings with neuroscience. So as I said, I'm from Ireland. I grew up in the town in the middle of the country, just right here, called Tullamore. And growing up, I wouldn't have been exposed to neuroscience at all. Neuroscience is quite a new field of science in Ireland in terms of funding and resources and research. And it is mostly kind of focused on the main cities, so Dublin or the south of the country, Cork, which I didn't live too close to either of them. So I wasn't aware that it was a degree course or an option to me until I was about 17 or 18. So I originally became interested in it because as I began to research what I wanted to do in college, I liked the idea of psychology and thought that I wanted to be a psychologist. Because I was really interested in human behavior and how humans interacted and the nature versus nurture debate. So environment and genetics and how that has a role on the development of a person and how they behave and how they act. And as I began to explore the options that were available to me for college, I realized that I was trending more and more towards the underlying biology aspect of psychology in terms of what role the underlying brain circuitry and the genetics had on determining the way somebody thought or behaved. And I was constantly trying to come up with theories or reasons as to what would be affecting somebody's behavior. So, for example, one thing that I always try to wrap my head around and come up with ideas about was how it is, to give an example, that two soldiers can go to war and experience the same environment, experience the same trauma, the psychological trauma, and be involved in like combat and dangerous and awful scenarios that you don't even want to imagine. But they finish their term and they come back or whatever to their own environment, where they grew up or where they live or whatever. And how it was that maybe only one of them might have developed PTSD, so post-traumatic stress disorder, which is a psychological condition characterized by overwhelming sense of fear from the trauma that would have been experienced in that previous example that I was explaining in a combat environment. So I was really fortunate to have an excellent biology teacher in my final 2 years of school who really promoted science and was very encouraging. And she was definitely instrumental in me choosing neuroscience over psychology because she had a very systematic and direct approach to experimentation and to carrying out kind of research type activities. And she was definitely an influence on my scientific learning journey. And there were a few people kind of over my, I guess, development of my scientific interests that were really instrumental in shaping the way that I went. And yeah, that's something that's really important. So going on to college then. So the college system in Ireland is presumably so very different to here in the US. And without wanting to go into too much detail, you apply to a college course, which is essentially the name that we have for what the US system would call a major. and as opposed to applying directly to a college. So you fill out a document listing about 8 or 10 majors that you would like to study in descending order of preference. And then depending on what your final exam results are from school, you are given... So depending on what your results are from school, from your exams, you're awarded an amount of points that ascertains to your results. And then the courses that you had previously listed on that document are also given a points level based on the interest for that course and the number of spaces that are available. And then you are awarded the highest course on your level of preference that you have sufficient points for. So I knew from the time that I applied for college that I wanted to do neuroscience, but this major was actually a specialization that was included within a biology course called Biological, Biomedical and Biomolecular Science. So I applied to that course and luckily got my first choice to study this course in University College Dublin in Ireland. And then the first year I covered mostly general biology courses with some chemistry and physics kind of thrown in just to get a general overview. And then in my second year, I could narrow my choices down to 4 subject areas. So I chose neuroscience, genetics, physiology, and microbiology. And then for my final 2 years, I narrowed down my choice even more to just focus on neuroscience. So after the 4 years, I graduated in September last year, September 2019, with a BSc neuroscience degree. And then as part of my curriculum for my final year, I got to carry out a research project and write up a thesis of the work I did. So I did my research in the lab of Dr. Gerard Cagney, who is an excellent PI in the biochemistry division of the Conway Research Centre in UCD. And I worked on determining a protein interaction map of the SETD1A protein, which has been found to be implicated in potentially causing, but at least largely contributing to some cases of schizophrenia, which, as I'm sure some people might be familiar, is a psychiatric disease that's characterized by hallucinations, delusions, disorganized thoughts, disorganized behavior, as well as other symptoms. And I found the project really enjoyable. I loved being in the lab. And despite it being a biochemistry-based project, I loved the link to psychiatric disease. I was so intrigued to try and figure out what this protein interacted with in the cell, which is hoped might give us an insight into what the protein itself does and how its incorrect functioning might actually contribute to schizophrenia. So I then wrote up my thesis document, which is about 10,000 painstaking words, and I have a picture of it there inserted. Very proud piece of paper. And I presented my work then to my classmates and other PIs within the School of Biological Sciences. And then I finished up with my exams, and along with my thesis, I was awarded my degree, as I mentioned, last September. So moving on to the fun part of where I currently work and what I do. So I am officially a technical specialist, but essentially just a scientist in a company called Neurophotometrics, which is a fiber photometry company. I will go into that a bit more of the scientific detail of what that means in a minute. But essentially, we are a biotechnology company that designs and develops microscope-like systems for use in neuroscience research that is sold to neuroscientists and companies in research institutes and settings all over the world. So Neurophotometrics was set up by Dr. Sage Aronson in, I think it was around 2018, just as he was coming to the final stages of finishing up his PhD. And for his own PhD project, Sage was using fiber photometry as an experimental technique. And he was tasked with the painstaking job of building his own fiber photometry system to carry out his experiments. And when he realized the difficulty and the expense involved in doing this, he had the idea to set up a company building these systems so that other researchers and poor PhD students like himself didn't have to spend the same time, energy, and resources building such a complicated and a complex piece of equipment. So since then, the company has gone from strength to strength, and we are currently getting ready to launch the newest version of our system, which has been much improved since the first iteration and has many improvements on the system that Sage would have originally developed a couple of years ago at this point. So, of course, COVID, like with a lot of other industry, interrupted a lot of what we were doing and interrupted a lot of the daily running of the company and the development of the system. So there was a brief transitionary period whereby we were making cotton-tipped applicators for use in COVID testing, and this was the Swab 56 project. I don't have time to go into this right now, but I have the logo there present on the screen. If you want to look into it, if you Google, you'll find some more information. So in terms then of what I do and work on a day-to-day basis. So a typical day at Neurophotometrics starts for me at 7 a.m. here in San Diego. I usually start by having my temperature taken in these wild COVID times. And then this is usually followed by a brief discussion with my boss, Veronica, and my colleagues about where we are in terms of production and development and what the tasks are that need to be carried out that day. So the components of our system are built using mostly using 3D printers. So I usually spend a lot of time during the day getting the printers ready, refilling the material, taking off builds and inspecting them, followed by cleaning them and getting them ready to go into the system. So during a production run, I spend a lot of my time building systems. So when I decided to study neuroscience, I thought I was going to be spending a lot of time around cells or brains or animals even, but I actually spend the majority of my time with a screw gun in my hand or a hammer, hammering things in. So that was definitely a change when I started, but it's something I enjoy. So I also put in optics into our system, which are essentially small pieces of glass and some mirrors that fit into the system to make sure that it functions correctly. So it is an optical based system and works off different physical properties of light. So it requires the optics themselves and the mirrors to make sure that there's correct kind of reflection and wavelengths passing through the system and all that kind of thing. And then once the system is built itself, it has to undergo a QC procedure, so quality control check. And when you bear in mind that this system is being used in research and it's being used to record very minute changes, it gives you an idea of how precisely the system has to be built and that everything has to be functioning optimally in the system itself to ensure that it's able to correctly acquire and collect data that is going to be eventually published and hopefully will make a difference to the neuroscience community and neuroscience research in general. So pre-COVID, we also carried out installs, which are when we would travel to a lab that has bought our system, and we would show the researchers and the students how to use it, and we would set it up in their lab. We plug it into any animals that they have ready, and we're able to get a sense of the research that they're doing and see a little bit of that. So this was one of my favorite parts of the job. And I would also spend a good portion of my day answering technical support questions for people that have our systems. So I'd spend quite a good bit of time through email or over the phone or online, kind of explaining if it's a thing that somebody's system is slightly off kilter or something kind of isn't working as it should be. I spend a lot of time online trying to fix that with them as well. So oftentimes the type of job or career path that is associated with studying any type of science is academia. But this position has really opened my eyes to the number of other avenues that are available. So my long-term goal is, in fact, to carry out research in an academic setting. But I'm really, really glad that I've had the opportunity to try something a bit different and to learn the skills that come with this position that I definitely will be able to apply to many other types of positions or challenges that I could face in the future. This job also, in my opinion, kind of blurs the lines between what is often strictly seen as two separate things, of academia versus working in industry. And the job features an overlap of both of these areas, since we are working within a company structure, but we still get to work very closely with the researchers, and we get a first-hand insight into their new discoveries with such a state-of-the-art experimental technique. So because it is also such a niche and new area, it's really nice to see that it is possible to work within an industry classification without it being a pharmaceutical company, which is usually what people think of when they think of scientific industry, I guess. And then finally, another real advantage that has a bit less to do with the scientific element, but that is just as important, is also the team of people that I work with. So my colleagues all have either an undergraduate or a PhD degree in neuroscience, which means that we all come from the same scientific understanding background, and we are involved in the day-to-day runnings of the business as scientists, as opposed to coming from a business background or sales background or marketing or whatever. We are a really close-knit group, and we get on very well together, which definitely helps when you're spending so much time with people, Monday to Friday, 8 hours a day. I'm also really, really lucky to have my incredible bosses, Sage and Veronica, who is the COO. I'm really lucky to have them as resources for any questions I may have on like scientific concepts or on aspects of like career development, since both of them have got their PhD, and Veronica also has 2 postdocs. So they're very helpful to go to and are definitely a good network to have in terms of being able to ask somebody with more experience about a certain thing, whether it be applying for PhDs or how to correctly fill out your application for a PhD or those kind of things. So the science then of the system and how it works. So the system works to measure levels of fluorescence or light that is emitted by activated neurons within the brain. And of course, as you can see in this picture, which indicates that this is carried out in animal models. So the protein that is responsible for producing the light that is measured by the system is injected into an animal's brain using a virus, and it's injected into the region of interest in the brain. This virus incorporates the instructions of how to produce this protein in the neuronal cells. So this protein does not constantly glow, but it only produces light when the neuron it is present in is activated or is active, and when a light of a certain color is shown on that area of the brain through a fiber connected to a wire. So the light that is then emitted from the glowing protein within the brain, within the activated neuron, then travels back up through the same wire, where it is detected by a camera and recorded by a computer. And this data then can be analyzed, whereby high levels of fluorescence indicate a lot of neuronal activity, and consequently low levels of fluorescence indicate low neuronal activity. This can tell us a lot about how the brain functions, and the outcomes of this research can be applied to better understanding how the brain functions correctly, as well as being applied to what happens in disease processes in the fight to help cure or prevent them, or just to even understand them a little bit better. So my plan for the future, as I've alluded to at an earlier point in the talk, my plan is to apply for a PhD. I plan to return home to Europe to do this, and I hope to start my PhD in September 2021. I'm still trying to decide on what exact type of research I would like to do, since I'm quite fortunate to have such a wide range of interests, but this can sometimes leave me with too much choice and make it difficult to commit to one thing to study. So I'm currently looking into human research, which would be more of a cognitive neuroscience stance, But I also know that I like animal research. But I think overall, I'd be happy once there is an underpinning of the research involved in a focus on psychiatric disease processes. So longer term then, I hope to do a postdoc following my PhD, and to eventually set up my own lab within a university or an academic setting, and to carry out research and to teach and lecture. But I have to say, like I mentioned earlier, the job that I currently work in has definitely given me much food for thought, and has promoted the idea that there are other available avenues that you can work in terms of science and neuroscience. And it has really shown me that you can definitely find a position that molds multiple aspects of the things that you like about science together. You just have to be willing to look for that position. And like Sage, if it's a thing that you see a gap in the market, you can more often than not set up your own company or basically establish your own job that suits exactly what it is that you enjoy about what you do. So I think this is something that's important when speaking to high school students or to anybody that might be a little bit younger than me, that I was once in their shoes and I was once in their position. And basically the kind of advice that I would have liked to have heard when I was at that stage of my career, I guess you could call it. So this one kind of goes without saying, don't ever let anyone tell you that you can't do it, and that includes yourself. So this gets thrown around a lot, and for good reason. And it's advice for so many aspects of life, not just career. But people hear this and think that it is going to be very obvious to identify if somebody says those words outright to them. But more often than not, that's not the case. Somebody doesn't turn around to you and say, you can't do it, or you won't be able to do that. Often they say it a bit more subtly, such as a quick comment in passing by, that is a lot of times carried out by an adult in a higher position, or a teacher, or sometimes even a parent, and comes in the form of, oh, but it's such a long path, or oh, it's so expensive, or oh, that's going to be very difficult, or it's a long road. And a lot of times people say it coming from a good place, but they don't realize that what they're actually doing is discouraging. And if you want to do something, decide right now and commit to it. Tell yourself that you're going to do it, convince yourself that you're going to do it, and don't let anybody change your mind. But on the same point, if you do change your mind for your own reasons, and you do want to change your own plan, know that you're doing that for yourself and for your best reasons, because life gets in the way and sometimes plans change along the way. So be willing to not listen to yourself when you're telling yourself you're not good enough, but also know that should you change your mind or should you not want to do that anymore, it doesn't mean that you failed. So the next one, life happens and gets in the way. Don't sacrifice that either. So this point might be equally as important, and it is definitely not discussed enough in terms of career development, and especially in science, I think. But it is the importance of also having a life. So, when you're in school, and I'm saying this as someone who was in that position in school where my study meant everything to me, and it was my number one priority, and a lot of what you focus on at that age is school. And even in college, this was the case for me when my education was probably still my number one priority. But you have to remember there are so many aspects of your life that you have to consider and juggle. And your career and your education is probably not always going to be as much of a priority as it is now. Unexpected things happen in life. It doesn't always go as planned. But it's very important to have the mindset and perspective that at the end of the day, your test tubes or your stem cell culture isn't going to be sitting at home waiting for you at the end of the day to ask you if you're okay and comfort you when you've had a bad day or you're feeling lonely or you're feeling sick or whatever it might be. So things other than your career will make you happy too. So you have to make time for them and take the time to look after yourself and what's best for you too. And then finally, there is no best path. Just pick the one that makes you happiest and gets you a step closer to where you need to be. So this one is potentially the most important one for me, especially at the minute. because I found from a very young age, I always questioned myself on this one, and I still do it to this day. So you're always going to think, oh, well, which is the best option? Or you're always going to be faced with choices, and you're always going to have to sacrifice something. But the thing is, if you have the self-reflective properties to question and to ask yourself, and put yourself in a position where you're really thinking about which of these options is the best for me to get me to where I want to go, to be the best that I can be. If you have the skills to do that, chances are you have the skills to make any option work, and to work for you. So especially now when I'm facing applying for PhDs, like I mentioned, there's almost too much choice. And I'm so worried that I'm going to pick the wrong one or pick the wrong PhD, make the wrong decision. But the thing is, all the choices that I'm being faced with are favorable ones. They're all ones that I like the sounds of. So I just need to get on with it. I just need to choose. And then after that, I'm going to work as hard as I can and do my best in that, no matter what it is. And that goes with every decision. I'm sure I'll be faced with that decision again at other points as well in my career. So this brings me to the end of my talk. I'd like to thank Chris and the Helyx Initiative for giving me this opportunity. I think the work they're doing is really good, and I would have really appreciated something when I was at high school age or going into college that is like this. And yeah, I've attached my email address here in case anybody wants to contact me directly, and I'd be more than happy to take some questions.
Well, thank you so much. Our first question is, your talk is very encouraging. Can you tell more about nature versus nurture?
Yeah, so this is something that I'm hoping to explore and potentially go into in terms of my PhD. But when people talk about nature versus nurture, they're talking about, I guess that's the colloquial term for environment versus genetics. So why is it that, like I said, the example of the two soldiers that go to war, they're both in the same environment in terms of war and psychological trauma and seeing combat or whatever it may be. But it is not always guaranteed that the two of those soldiers are going to come back and have PTSD. So is that something genetic? Is there a genetic underpinning that is, I guess, protective for the soldier that doesn't develop the PTSD, and that's detrimental to the soldier that does? I guess if you are really interested in something like this and you want to look into it, the major kind of thing that can't be ignored in the nature versus nurture debate is twin studies. So identical twin studies are used in studies for loads of different neurological diseases or psychiatric diseases in particular. So to give just a little bit of, I know a little bit about schizophrenia. So basically identical twin studies are carried out such that identical twins obviously have the same genetic makeup. So they have the same genes, the same DNA, and they have it in all of their cells, their body, and both of them are genetically identical. So it would be thought that since they have the same basic DNA blueprint, that the two of these individuals should be exactly the same. But I'm sure many of us know a set of identical twins, and usually one can be more sporty, or one's more artsy, or one is really good at running and another one doesn't like sport or whatever, so there are many differences. And the same comes down to psychiatric disease. So a lot of times schizophrenia has been found to have a genetic component, because if one identical twin has schizophrenia, the odds are very, very high for the other twin to also develop schizophrenia. But it's not always 100%. So it's not always a 100% given that if I develop schizophrenia, that my identical twin will too. So that means that there is an interplay between the genetics and the environment, or the nature versus nurture. So the environment could be anything from what they were exposed to in utero. So while their mother was pregnant with them, whether she contracted an infection, or she had some sort of like trauma. It could be anything, it could be nutritional. So that's one aspect of environment. Environment is also in terms of if it's a thing that those identical twins were then separated, and one was brought up in a safe environment where they were supported by their parents, and another twin was brought up in a damaging environment, that was completely different. So yeah, God, nature versus nurture is something I could go on about all day long. But if you want to get a really good idea, there's a documentary that I'm pretty sure is easily enough found through Google called 3 Identical Strangers, that will outline everything that you need to know basically about the nature versus nurture debate. And it's basically about 3 identical triplets that were separated for a research project. I think maybe like 50 or 60 years ago at this stage. So look into that, because that's it's definitely a good one. It's very interesting just from a general viewpoint as well.
Cool. So our next question is, I'm really interested in your thesis paper. How did you connect the role of SETD1A and schizophrenia?
Okay, that's a great question. So going into the project, I knew that SETD1A had been implicated in schizophrenia. So two research papers have been studied have been released in 2014 and 2018, where they did a massive study on thousands of schizophrenia patients, and they basically genotyped all of them. So they looked at their DNA, they looked at their genetic makeup, and they were basically able to identify that in a very, very, very small subset of those patients that had schizophrenia, some of them had a mutation, or a specific type of mutation, in the SETD1A gene. So SETD1A is a protein that is a histone methyltransferase, so it's an epigenetic protein. So basically it is involved in either in unbinding coiled DNA and making the DNA and the genes open to being transcribed and translated for expression of those proteins. So SETD1A basically controls kind of what genes are allowed to be expressed within the cell. So it's basically kind of like a regulator of the genetic activity. And so those studies have been conducted, and they basically found that people with this dysfunctional form of SETD1A were very, very likely to have schizophrenia. And there are very few genetic indicators that kind of give that level of certainty for schizophrenia, because it is so kind of on the border of like being genetically but also environmentally implicated. So I basically then was, as part of my research project, I was trying to find which proteins within the cell this SETD1A was actually interacting with. So I basically pulled down this protein. I was able to isolate it from cells, and then I basically used a mass spectrometry machine to kind of look and see which other proteins had bound to this protein. And then by a matter of deduction, I was able to say, well, if it interacted with this kind of protein, then it must be involved in this kind of cellular pathway. And if it bound to this kind of protein, it must have been involved in this cellular pathway. And basically, one of the most interesting results of my project that I figured out through pulling down that protein and seeing what it interacted with was that SETD1A actually had a very large interaction profile with components of the immune system. And this was really, really interesting, because more and more work is being done on the interplay between the nervous system and the immune system. And for a long time, people thought that the brain and the nervous system were isolated from the rest of the immune system, and that there was no kind of immune function within the central nervous system, due to the blood-brain barrier basically having such a tight kind of connection and protecting it from the periphery. But in reality, that's not the case. There are certain kind of CNS specific kind of immune regulatory systems, and basically the proteins that I found to interact with SETD1A are called complement. So they're proteinaceous components of the immune system. And why this is really important is that it is thought that mothers, while pregnant with a baby, if they are immune compromised or if they suffer certain types of infections, basically those infections or the immune response that is mounted to those infections can implicate the developing fetus or the developing baby. So I guess the hypothesis or the theory would be that these patients who have this mutation in SETD1A could potentially have been compromised by the upregulation of the immune response in the patient's mother when she was pregnant with them, and that this might have caused some sort of alteration in SETD1A, and then because SETD1A was compromised then, that could have been what contributed to the person developing schizophrenia. Hope that makes sense. That was a very long-winded kind of explanation.
No, that was great. Another quick question was, what was your favorite class in high school and why?
So this is another great question. I was actually going to put this in my talk, but I just wasn't going to have enough time. But when I was in secondary school, which is like the equivalent or whatever of high school, I remember like people talking about their favorite subject, and I remember thinking, oh my God, how do people have a favorite subject? I don't see how that makes sense. Like, I'd much rather be riding my horse than be in school and be doing this. Like, I'm doing this because I have to, because I have to go to college, I have to get a job, as opposed to really wanting to. And then when I actually realized kind of that just to like a subject, you don't have to be obsessed with it. It doesn't have to be the only thing that you ever think about. I realized that I had always had an affinity for biology. So my mom is a nurse, and she would come home from work, and I would say to her, like, what happened today, or what did you see today? And I can remember doing that since I was like a kid, and she would say something, or she'd explain, if we fell, she'd explain like, oh, your knee hurts because you've hurt this, or you're bleeding because it's this, or your blood is red because of the hemoglobin that's this, or whatever. And I was never happy with her just giving me one answer. I had to ask more. And it was like, oh, your blood's red because there's this thing in it called hemoglobin. And I was like, well, what's hemoglobin? And she'd explain a bit more. And I'd be like, well, why does that make it red? And when I realized that that's what it meant to like a subject and what it meant to like something, I realized that I was obsessed with biology. And it took me a very long time to come to that conclusion. So I had like an hour or two of biology class first thing on a Monday morning, my final 2 years of school. And, I remember like people didn't like science. Science was difficult, or it wasn't attractive, or it was like the nerdy subject or that kind of thing. And I realized I was kind of thinking differently from everybody in terms of that when I used to get excited to go to school on a Monday because it meant I had biology. And like I mentioned as well, my biology teacher, Miss O'Doherty, she was fantastic. And she really made me enjoy the subject. And I think she really promoted it in me because she could see that I had affinity for it, an affinity. And then because of that kind of promotion and that kind of encouragement from her, that heightened my my awareness and my love of biology for sure. Yeah.
Cool. So our next question is, when you were still in school, did all science come easily to you?
No. No, definitely not. biology did, just because I loved it, and because I always wanted to know more. And when I realized that it was the memorizing that I didn't like about it. I loved learning. I didn't like having to sit down and reread the same thing 5 times over until I could recite it myself. So biology came easy. I also did chemistry, and I definitely found that more difficult. And I think it was because, to me, it was so kind of abstract. It was something that I couldn't see. It was something that I couldn't kind of watch it happening. Like, if you cut yourself, you can see your blood coming out. Or like, if you see, I always think of that. I don't know if anybody ever has seen it. It's a very old movie, an old book, but To Kill a Mockingbird. There's basically a scene in that movie where there's a dog, and the dog has rabies, and the dog is like falling all over the place and can't stand up properly, and he's not moving properly. So like, to me, that was like, that's the brain working. The brain's doing something wrong. So you can see that. Whereas trying to tell me what a polar compound was and why water was polar but oil wasn't, that went kind of way over my head. So I chose chemistry. I could do chemistry for my Leaving Cert, which is the set of exams at the end of school. But I did kind of question whether I should have stuck with it or whether I should have taken something different. But I realized then it is helpful to have that, because especially if you stay in science, I obviously stayed with neuroscience, but it is helpful to know those kind of chemistry basics. So answering that directly, no, it didn't come easy. Chemistry didn't come easy, but it definitely still helps to this day with the kind of stuff that I'm doing.
We are a bit over time, but hopefully, if you can stay, we have a few more questions.
I can run a bit quicker through them.
Yeah. Claire says, have you seen a trend that more girls are choosing STEM?
Yes, definitely. I mean, so I graduated from school 5 years ago, and even since then, when I was in my final stages of school, I could even see in Ireland, in my school, in my little town, that more people were coming in and more people were encouraging and promoting STEM and kind of highlighting why it's so important, and especially with females. So yeah, it definitely, more people are choosing it. It's becoming more attractive. I think there is like still it's in particular in like academia, it's very elitist. It's very white male dominated. But that is definitely changing. And I think, like was said earlier on as well, kind of programs like this are definitely helpful and are really promoting it. And there's less barriers. The more people that get involved, the less barriers there are, and the more people that talk about it and continue to raise awareness and ask those types of questions mean that it's always going to grow and it's always going to be more encouraged. Okay. So the last question, can you expand on how viruses were used to insert proteins to detect light emitted by neurons? This is a great question. Okay, I'm going to try and do this as simple as possible, because these kind of concepts can get a bit complicated, and I was trying to make sure that it was all inclusive, but great question. So basically, these viruses are termed vectors. So they're basically made into inactivated viruses. So they have a section of DNA basically put inside the virus. And what happens is the virus is injected into the part of the brain that you're interested in. So whether it's cortex or it's wherever, limbic system, subcortical, any of the subcortical areas, whatever. And then based on the virus, based on where it's injected, based on the type of DNA that's within the virus can target different parts of the brain, different cells, different types of cells, whether they're GABAergic or dopaminergic or whatever. So that targeting occurs, and then basically the virus invades the neurons that it's supposed to, or that it's able to. And the way viruses work is that they inject DNA into the cell, and then the machinery within the cell work to replicate that DNA, and it's incorporated into the DNA of the cell itself. And then that basically transcription and translation occur, and that gene is expressed, or in this case, that protein is made. So the most kind of common fluorophore protein is called GFP, green fluorescent protein. So basically this code of DNA is expressed within the cell, and GFP is made, and GFP stays within the cell. And then essentially when a neuron becomes activated, calcium comes into the cell, and calcium binds to the GFP. And when calcium binds, that's when the protein starts to fluoresce, and that's what's detected by the camera. And then once the neuron is no longer active, basically the calcium unbinds from the protein, and the protein then no longer fluoresces. So that lack of fluorescence is then indicated, or is I guess taken as showing that the cell is no longer active.
Yeah, thank you so much. I'm sure there's like a lot of questions that could be asked, but yeah, we are running out of time. But thank you so much for doing this presentation. It definitely shows a lot of people, especially like how the neuroscience career path may change, how the focus might shift. I'm especially like hearing that. I often get caught with like the philosophy and psychology part, and like I kind of get scared that going too much into chemistry or going into industry will be like kind of taking away from that aspect of it. But I think it really shows that you can be active in like multiple fields. But thank you so much to both of our speakers for coming, because I think Asome is still here. But yes, we had a really great time. If you guys have any like parting words to say, please feel free. And yeah, thank you to everyone who's coming to these talks as well. And hopefully we will see you next week. To answer Claire's question, we will be having another girls' night. I believe it's Girls' Medicine Night next week. But Sarah can.
Yeah, the Girls' Medicine Night is next week from 5 to 7 p.m. Pacific time.
Oh, there's still people here, but yeah, I really enjoyed your talk. Miss Deegan, I have to ask, since you're from Ireland, do you like James Joyce, or is that just?
This is funny. So I'm not familiar really with his work at all.
Okay.
The only reason I am familiar with James Joyce is because that is the name of our library in college. So the library in UCD is called James Joyce Library. So now that's all I associate with the name now. But yeah, unfortunately, I don't know much of his work.
I really like literature, so, and he's he's one of the famous University College Dublin like alums.
Yeah, for sure.
Thank you so much. Take care.
Thank you so much, both of you for coming. Yeah.
Thank you. Bye.
Bye.
Bye. Thank you.